Hello students, now we will start with new topic about MYCOTA, means fungus. In case of Protista, types of nutritions, we have seen are, first one, heterotrophic. Example is amoeba. Parasitic means by absorbing nutrients from the host. We see entamoeba, trypanosoma, etc. Next, autotrophic. Here we can see example paramecium. And also saprophytic. Example we see slime molds. Since in one group of Whitaker's 5 kingdom classification, we are seeing all different modes of nutrition. So we can use a term or a sentence here like that. Protista includes diverse group, nutritionally diverse group of organisms. So we can say Protista includes nutritionally diverse means different group of organisms. So this is about Protista. Next we will see about fungus. So fungus is studied under MYCOTA. That is study of fungus. That is mycology. So here we read completely about fungus. So fungus or MYCOTA, the terms are same. So what are characteristic features? First we have to remember that they are saprophytes. Cell wall is of chitin plus glycans. Chitin is chemically composed of N-acetyl glucosamine. Glycans includes 2-deoxyglucoses. These two different types of compositions constitute the cell wall of fungus. They are considered as close relatives to animals than to plants. So they do not have chlorophyll. So there is no chlorophyll. So since they are not completely depending on other organism, in the sense they are heterotrophs, but under heterotrophs specially they are read as saprophytes. Fungus reproduction is both by sexual and asexual mode. Under asexual mode, we can see budding and fragmentation. Under budding, we can see example like yeast, Saccharomyces cerevisiae. Under fragmentation, we can see example like agaricus, that is mushrooms. Now, under sexual mode of reproduction, we see meiosis forms meiospores. Now here there is no specially name for sexual mode of reproduction, but we can say sexual mode of reproduction is by plasmogamy. which is later followed by karyogamy, later followed by meiosis. So, the process will be first plasmogamy. This is followed by karyogamy. This is followed by meiosis. What is meant by plasmogamy? Fusion of protoplast of the chambers or the one hyphae or a part of hyphae. Fusion of protoplasm means whatever the nucleoplasm plus cytoplasm content, life content of the hyphae. Karyogamy means fusion of nuclei. Meiosis means reduction division, in which the diploid cell will undergo meiotic division to give haploid gametes. We'll see diagrammatically. This is aseptate hyphae. This will give mostly means asexual mode of reproduction by production of sporangiophores, chlamydophores, zoospores, etc. So, mostly they exhibit asexual reproduction. Now, if the hyphae is septate, here one nucleus, here one nucleus, here one nucleus. Now, another hyphae will try to fuse with this. We can say this is A hyphae, this is B hyphae. In the next stage, what happens? The chamber of this hyphae, they protrude towards each other and forms a connection like this. Now, this connection will be digested, and in the next stage, here forms two nuclei containing one single chamber. Now this one is termed as, this single one is termed as dikaryon stage. Later these both will be fused, that is karyogamy. After formation, it leads to zygote formation. That zygote now undergoes meiosis to give out meiospores. So, dikaryon stage is seen in the sexual mode of reproduction during the meiospores formation. Now, after meiosis, the structures formed are, the spores formed are haploid, and these are considered as single-celled propagules. Single-celled propagules. Why the term propagules? Because after formation of the spores, those spores get separated from the parent body and they get dispersed to some other area along with the wind. So such types of structures are called as propagules, which get separated from the mother's body or from the parent's body and get dispersed into some other area. The spores. Spores are single-celled haploid propagules. Now, we can give other term for propagules as are vegetative forms. They later gets separated from parent. We will see some types of spores. Example are zoospores. Zoospores are motile spores formed during asexual reproduction. This is one of the previous MCQ. Motile spores formed during asexual reproduction. Aplanospores, non-motile spores, again formed during asexual reproduction. Another type of spores are chlamydospores. These are of again zoospore type and also aplanospore type and also echinid forms. Why? Because basing on the conditions that are favorable or unfavorable, the type of spores in case of chlamydomonas differs. So can be of zoospores, aplanospores or echinids. Now again, spores formed during asexual reproduction. Sexual reproduction are ascospores, next basidiospores. So ascospores are formed in sac fungi. Basidiospores are formed in club fungi. We will see in detail about all these types during classification. Now we will see how the hyphae is formed from spores. I said you spores on germination gives hyphae. But this is not direct. There are some stages here. Spores are considered as haploid structures. They germinate to give, if this is the spore, it gives a thread-like structure. This is called as protonema. This protonema is considered as juvenile form, means newly formed one. Juvenile form. It represents newly formed gametophyte or juvenile gametophyte. So here we are seeing formation of protonema. This protonema later develops into hyphae. Now, one point regarding dikaryon. When during the sexual reproduction, when two different chambers of two different hyphae are fused, they form dikaryon stage. So we can say, if this is the dikaryon with two nuclei, means karyogamy did not happen, just plasmogamy. So dikaryon, this is after plasmogamy. At this stage, we can say it is also termed as heterokaryotic stage. It means, hetero means different. So different nuclei from different hyphae chambers formed into one single cell with two nuclei, so called as dikaryon. So also called as heterokaryotic stage. Later this protonema develops into hyphae. So from dikaryon stage is formed later the spores. So here we can see karyogamy happens, one single nuclei, means zygote-like structure. Later it will undergo meiosis and forms spores. Those spores on germination again gives rise to these type of structures.